Good morning, guten Morgen. Thank you very much for the opportunity to deliver this keynote lecture on computational and analytical methods for getting the most from your targeted proteomics experiment. And so I'm not going to be talking a lot necessarily about Skyline and the things that you can do there or about experimental design, but hopefully give you an idea of where you can take the data that you'll generate with all of your targeted assays that you'll be so good at configuring going forward. Again, I'm Jake Jaffe from the Broad Institute in Cambridge, Massachusetts, just in case there are some guests who are here today who were not here yesterday. And the talk will consist of 2 or 3 parts, depending on how we do on time. And again, I am happy to keep this as a casual lecture. People should feel free to interrupt with questions, and Ben or someone else from the course will bring around a microphone for you to ask your questions. So today I'm going to talk about targeted proteomics for generating what I call signatures, and using those signatures to formulate connectivity, or what that means, the concept of connectivity through signatures. Then I'll try to show you some real-life applications of generating connectivity maps from proteomic signature data, and thinking about new ways to query these signatures, not just looking at patterns on heat maps and things, but principled ways of using proteomics data to perform lookup and query functions like you would do almost in a database. And if there's time, which I don't think that there's going to be, but if there's time, I can talk a little bit about converting what is a PRM assay into a hybrid PRM DIA assay. And if any of you, if I don't get to it today, but you're still interested, I'd be happy to talk with you in the breaks about that. Okay, so I was very gratified to see Rudy's talk on the first day of the course, where he talked about kind of data, shapes, and proteomics, and I have quite a similar slide, and I think most of you are familiar with this paradigm where if you're doing shotgun or discovery type proteomics, you can end up with a couple different types of data shape. One is the kind of long, skinny shape. So let's imagine that every column here is a different sample, and every row is a unique protein or peptide, analyte. And so if you do just a few samples, a couple of samples, you can have lots of analytes that you see in common between those two samples. And maybe there's a couple of places where you see it in only one or the other sample. Or if you interrogate many samples, you can get this situation where there are only really a few analytes where you observe most of the proteins across the entire sample set. More typically, what we see in shotgun proteomics experiments is some relatively large number of samples, some large number of analytes, and lots of holes in this matrix. So I call this holy data. These are holy data. And in this case, being holy is not desirable. And one of the nice things about targeted proteomics data is that it tends to be unholy. So in this case, being unholy, at least from my perspective, is desirable. And this is just an illustration of this. These are data from, again, the histone assay that I introduced yesterday, where we have now many samples in the columns, and just I'm showing a subset of the analytes here, and you can see that there are very few holes or black spots or gray spots in this matrix. And so now we can really do something with this. We can do hierarchical clustering, which in this case we've done. I've just omitted the dendrograms here, and we can look very carefully at the patterns that emerge from the clusters or the groups that form from that. And in this particular case, what I'm showing you is about 100 different cell lines. So it's 100 different genotypes that are all cancer cell lines. And I think you can appreciate from this heat map that different cell lines seem to have different patterns of the analytes. It doesn't even matter what they are right now. The fact that there are different patterns that are emergent and reproducible among all of these samples, and they can be formulated into groups, is very nice. And I don't think we could have got this through a shotgun approach for these particular experiments, but having the unholy data through targeted proteomics has gotten us here. And so I want to talk a little bit about what I'm calling a signature, a proteomic signature. So this map is just a subset of the map on the prior slide, where I'm showing you a blow-up of the data from clusters A, D, and F. And here you can see even more clearly that among these groups of samples, there's a repeated pattern in the analytes. Some analytes, let's take this cluster A, some analytes here are down, some analytes are up, some are sort of intermediate, a subset of them might have that up, but we could distill from this a pattern or a signature that typifies this cluster. And then you can see here in cluster F, it has sort of a very different signature. Different analytes go up, different analytes go down. And so this cluster F has its own signature. And then cluster D, this middle cluster, also has its a unique signature. And it turns out that we can correlate these signatures to different genotypes in the following way. In this first cluster, all of these cell lines happen to have point mutations in the protein called EZH2, which, as I mentioned yesterday, is a catalytic methyltransferase for histone H3, lysine 27. And all of these particular point mutants fall in a given domain called the set domain. It's the catalytic domain of this enzyme. And what we can glean from looking at the signature and looking at the analytes that are upregulated, that these point mutants are in fact gain-of-function point mutants. We can see lots of increase on the marks that contain the trimethylation of H3K27 and downregulation of some of the lower states of methylation of H3K27. So if we skip over to this side over here, what we find is sort of the opposite kind of signature. Now, the lower states of methylation are upregulated. The higher states of methylation are downregulated. Many of the lines in this particular cluster have point mutations in the same domain, but these are now loss-of-function mutations. We're also able to infer about this line here, this KE37 line. It has a point mutation in a different part of EZH2, not the catalytic domain. But here, the signature strongly suggests that it is also a loss-of-function mutation as sort of a guilt by association with the other guys. And then there's this middle cluster here, and this has two very interesting genotypes in it. In all of the cell lines here that are annotated with a little black box here, they have a whole chromosome translocation that puts the gene NSD2 under a strong promoter, where it normally would not be under a strong promoter. So it's kind of like a gain of function for NSD2. And then we saw these other cell lines were co-clustering with this translocation event on these chromosomes. And it turns out that all of these lines, now annotated with the pink boxes, have a single point mutant in that same gene, NSD2. And we were able to recognize through the signature that this single point mutant, single amino acid change in this protein is in fact able to recapitulate the entire phenotype of a whole chromosome translocation that changes the behavior of this gene. So using, recognizing through the signature, through the specific set of changes that seem to be repeated across these two very different genotypes, we were able to figure out the biology of this and went on to show that, in fact, it is a gain-of-function mutation and is potentially a driver mutation for cancer, specifically ALL. And so the idea of a signature is just this repeated, recognized pattern across groups of samples in a targeted proteomics panel assay. So we can use these signatures to formulate what I like to call connectivity, and this is just sort of a cartoon version of what I would call connectivity. Let's say that we profile in a panel assay, and let's say this is a phosphosignaling panel assay, the responses of cells to drugs, to different drugs being introduced into the cells and treat the cells for a certain amount of time. In this case, let's say aspirin, a common painkiller, and we observe some pattern or some signature across many replicates. And then another painkiller, Tylenol, it has a different mechanism. We may see some other signature in the assay panel. And then we do some genetic experiments where we knock down a gene, and we've knocked down this gene here called PTGS2, and we notice that it has a similar pattern to, say, aspirin. what we can do is simply calculate the correlations. Well, first we can plot things on a heat map, but more importantly is we can calculate the correlations between the signature of the different drug painkillers and then the knockdown of this particular gene. And what we would find, which is probably pretty evident, is that the correlation between aspirin and knocking down this gene is much higher than, say, the correlation between Tylenol and either aspirin or having knocked down this gene. And it turns out that this gene that we have theoretically knocked down here is, in fact, something called the COX-2 inhibitor. It's the mechanism through which these NSAID drugs like aspirin or ibuprofen work. And so you would surmise that inhibiting this protein by a drug should generate a similar signature to having inhibited that protein by, in fact, removing it from the biological milieu. And then once we've calculated all of these correlations, we can visualize them in a very intuitive way by simply saying, let's take each of our conditions here and plot them as a node or a circle in a graph. And then we can link them up with lines, and we can say that the weight or the strength of this line is proportional to the degree of correlation between them. So this cartoon here that I'm showing you is simply these data with the correlations plotted in a visual way. And then it's very intuitive that the aspirin treatment is closer to knocking down PTGS2 than an either R to Tylenol. And so now let's say we do some new experiment where we take a new drug, a new painkiller candidate. We want to ask the question, how is this thing working now in cells? So we can use our targeted assay panel to generate new data. We simply add it into our heat map and calculate further correlations. And again, it's already apparent that maybe this painkiller has a signature that's more similar to aspirin than Tylenol. But again, representing it in this connectivity map form by using edge weights that are proportional to correlation, and then allowing the graph to kind of self-organize using a spring-embedded, force-directed model, it's very intuitive that things that come close together behave in similar ways with respect to the targeted assay panel. They have similar signatures in the assay. And so that's what we mean when we're talking about a connectivity map. And today I'll be talking about some proteomic connectivity maps that we have generated and tell you a little bit about a project that we've been working on. Okay, so I'm going to talk about two different panels. One is this chromatin modification assay panel that I introduced briefly yesterday, and we call this, for shorthand, we call it GCP, or Global Chromatin Profiling. And I'll tell you about a second assay, which is a phosphosignaling assay panel, which we call P100. And this takes a kind of reductionist approach to phosphosignaling that I hope to share with you. And so yesterday we talked about what this assay is reading out and that sort of biology that it's reading out. And it turns out that we know that there are associations with epigenetic processes and disease, but there are associations with all kinds of environmental conditions in epigenetics. It's potentially a metabolic sensor. Epigenetics have been proposed to be a readout of cell metabolism and nutrients that are available to cells. So it's really quite a catch-all assay, and lots of different sorts of conditions or treatments that we might do to cells could have an effect in the epigenetic space or in the chromatin space. So that's why it's one of our favorite assays. And I also showed you this slide yesterday, but the thing that I want to point out today is that through our robust processing pipeline, we're doing all of these operations 96 samples at a time. So we do things at the level of a 96 well plate. The number of cells that we grow for each of these assays is more than you can fit in a 96 well plate, but when we pool everything all together, we try to do 96 at a time. And so we've relied heavily on automation. Here we do a lot of our biochemistry using automated liquid handlers. We try to use 96-well extraction devices for cleanup when we can do that, and so on and so forth. And it's really made our pipeline quite reproducible as well as robust, and gets us to a higher throughput. And I think generally the folks working in the lab are much happier when the automated liquid handling is doing all the pipetting rather than they are. So there's that. Okay. Yeah, so that we already know. So now here are some early data that we generated using this chromatin assay panel, where what I showed before as a cartoon is now sort of has an example in real life where we not only knocked down genes that we knew were important for epigenetic processes, but we also treated with drugs that we thought would inhibit epigenetic processes, and then made total genetic knockouts of certain enzymes in epigenetics. So again, what you can see here across the top are the names of the genes that we knocked down, and we used at least 2 or 3 different small hairpin RNAs to knock down those genes. So we have kind of biological replicates, technical replicates. Well, they're actually all biological replicates here, but we have different ways of knocking down the gene. So we make sure that no matter how we get there, we kind of get to the similar answers. And similarly here, we have knocked down some things in the G9A machinery. All the knockdowns are encoded with little gray boxes below. And then we also treated with drugs, which are encoded with these yellow boxes, or we made genetic knockouts were encoded with this red boxes. And then, of course, we did lots of controls, scrambled hairpins, or in the case of drug treatments, we just treated the cells with vehicle. And so what you can see first looking at this control area is that most of the controls here have quite a neutral effect on all of the histone marks in the panel, which is what we would expect. And then, in fact, there's one knockdown reagent that we use, the second small hairpin against a gene called EED, that also seemed to have sort of a neutral effect and didn't do much in the assay. Turns out when we went back and measured the knockdown efficiency of that gene, or of that hairpin, in fact, it did not knock down the gene very well at all. So in a way, it kind of serves as a control, the exception that proves the rule. And it was correctly grouped with its controls. But then you can see the very strong signatures in the assay, whether we have perturbed this G9A system by knockdown, drug, or knockout, or have perturbed this polycomb repressive complex 2 system, again, by knockout, drug, or knockdown, they all generate very similar signatures. So we can collapse all these replicates, all the replicates for each of the hairpins that we used in the study, and formulate them as a connectivity map. And what you get is this. We can see that all of the unique hairpins against PRC2, except for one that was more of a control type condition, all self-organized together in this map. And the way I've encoded these is the inner color of the circle represents the sort of biology that we're targeting with the particular perturbation. The outer circle represents the type of treatment it was, whether it was a knockdown, knockout, or a drug treatment. And so here you can see all of these green nodes have self-organized together, regardless of whether it was a knockout with the gold rings around or whether it was a drug treatment with this pink ring around. And similarly, we can see in G9A, all of the G9A perturbations self-organized together. We can see that a drug that is targeted against G9A, in fact, organizes very close, and the knockout also self-organizes together with these perturbations. And so we can put now together on the same framework drugs, genetic mutants, and all sorts of perturbations, as you can imagine. And the other thing that's important to say is that these samples were done weeks or even months apart. But because the assay is standardized in such a way, we're able to kind of put everything together on the same ruler of the targeted proteomics assay panel. And in fact, those of you in the course, you've actually worked with data from this study when we did the quick walkthrough yesterday. So let me now tell you about what I call a reduced representation phosphosignaling panel, or something that we call P100. And so here's the premise behind it. As I said, that we think that we can use a reductionist approach to kind of get a general sense of what's going on in phosphosignaling. So the premise is this: phosphosignaling is likely to be coordinated in cells. There are probably on the order of 1,000 kinase and phosphatase genes, but anyone who's done any phosphoproteomics knows that there are in excess of 100,000 different phosphosites that have been mapped in the human proteome, and you can go on to phosphosite.org and download all of these. So by that very fact, it must be that one kinase must be able to phosphorylate many substrates or many different targets. And so you might imagine something like this. You have a kinase that phosphorylates many targets in a coordinated way. One of those targets may in fact be a kinase itself, which goes on to phosphorylate many substrates, again, in a coordinated way. And so the question we started with is, do we really need to monitor all 100,000 phosphosites in the phosphoproteome all the time to understand what's going on in phosphosignaling? Or is there some kind of representative set, a reduced representation set, or a proxy set that we could use just in order to get a general kind of gestalt view of what's going on with phosphosignaling? And so that's where our reductionist approach was born. I'll say that we were inspired to do this by a similar assay that had been configured at our institute in the space of transcriptional profiling. There's an assay there called the L1000, where they take all 20,000 potential transcripts and have reduced that to a space of about 1,000 transcripts that they can monitor very cheaply and at high throughput. And we said, well, we can probably do the same thing in proteomics. And so the P100 idea was born. And the way that we got there was by collecting a lot of traditional proteomics data, shotgun proteomics data. We took 3 different cell lines. We treated them with 26 different drugs in biological duplicate. We did this weeks apart and had a giant data set, 156 different samples, which was, of course, wholly, as I described earlier, lots of holes in the data. But importantly, there were over 1,000 sites, maybe between 1,000 and 1,500 sites, that we could see in at least 3/4 of the samples that we profiled. So this is a good place to kind of start. And we used those highly represented sites, and we condensed this down into our P100 assay. So the P100 assay has about 96 or around 100 different phosphopeptide probes in it, and is a more compact, easy to execute assay than looking for these 1,000 things in a shotgun proteomics way. Okay. And so the other important thing about these probes is that they're easy to observe. As I mentioned, we did the discovery in several different cell types, so we know that they can be found easily. They tend to be the higher abundance phosphopeptides, but we also know that these things change in response to perturbation by chemicals. And so we actually filtered out those kinds of phosphosites that did not change across many different drug perturbations. And what we're left with is something that we think will be a good readout of chemical or genetic manipulations of cells in general. But if we look back at the source data from which these phosphosites were drawn, not just the probes, but the entire set, we can show that they're mapped onto various pathways that are of biological note or biological importance. And therefore, we think that the assay should be able to project onto all of the pathways that I have depicted here. And I'm sure there's some favorites there, like insulin signaling, MAP kinase signaling, HER2 signaling, mTOR signaling, et cetera. And again, we have configured a highly automated assay workflow so that we can do these things 96 at a time. And in fact, we've even automated the process of phosphopeptide enrichment using some custom and now commercially available IMAC tips that we work together with Agilent to develop for their Bravo assay map platform. Okay. So without telling you all the kind of history of this and how it is, I just want to assert a few things about the P100 assay and make a short story here. So the P100 consists of a set of phosphosites that almost always can be observed in various cell types. They behave in interesting ways in response to perturbation. They give you information that you could not necessarily get from gene expression. And we've shown this in a sort of computational way, but I ask you to take my word for it for now. And we're able to condense down some larger set of phosphosites to be read out by a smaller set of informative phosphosites. And then, importantly, we were able to show that repeating the profiling using now only the targeted assay panel of our 96 phosphopeptide probes is able to recapitulate the sort of observations and conclusions that we're able to get from doing the thousands of phosphosites that we monitored in the initial discovery experiments in a very short time span relative to what it takes to do a shotgun proteomics experiment of that size. So rather than gaining data in the matter of months, we were able to recapitulate all of those data through the panel in the matter of weeks. And then finally, the assay is generally useful. It's not overtrained to just the conditions that we developed the assay on, and I'll show you a few examples of that. Okay, so the one quick example is to look at response in the assay to the very pleiotropic kinase inhibitor staurosporin. Staurosporin is known to inhibit many, many kinases, and what you would expect from staurosporin is that phosphorylation in general will go down in cells. And so what I'm showing you here are two different cell types that we've treated with staurosporin. We treated them in a dose-response curve anywhere from half micromolar up to 20 micromolar, which is very deleterious to cells. And then just clustered, hierarchical clustering as before, and you can see that the clusters sort of arrange themselves in a dose-response kind of fashion without us really needing to do anything here. And what you'll notice, I'm only showing you a subset of sites that are present in the assay at the moment. There are a set of sites, what I've shown here with number 1, that have the expected behavior that you would think staurosporin would cause, and sites go down, or the level of those sites goes down as the concentration of staurosporin increases. But we have a whole other set of sites that have the opposite behavior. They actually go up in response to staurosporin. And so this was a good early test for us that we didn't pick sites for the phosphoassay panel that just all did the same thing and all read out the same kind of biology. We're able to get differing responses here, sites that go down, sites that go up, and then there are a whole set of sites I'm showing you here that stay neutral in the presence of staurosporin. And these sites will have very different behavior in response to other different perturbations. Okay. Now, we can also take these phosphoproteomic panel data and arrange them into connectivity maps. And so now what I'm showing you is an arrangement of all of our initial experiments. Again, just using correlation coefficients to weight edges between these nodes, the way that the nodes are encoded. The center color is the class of drug that is being treated onto the cells here. The outer circle is the type of cell that we have treated with these drugs, and then we just let everything organize. And so what you'll see here is a series of blue nodes in the center, and these are all very related family members of a set of compounds called the cardiac glycosides. These are digitalis is the common name of the drug that someone might take if they had a cardiovascular disease, but a whole set of related compounds. So this is good. We can take structural analogs of one another. They generate similar signatures in cells, and then in a connectivity map fashion, they self-organize together. And importantly, they self-organize regardless of cell type. Here we can show that whether you're in a breast cancer cell with the dark blue rings or a prostate cancer cell with the light blue rings, the signatures are similar enough that these things will co-organize in a connectivity map. And then similarly over here in a different cell type here now, HL60, which is not a solid tumor, we can see that again, the cardiac glycosides organize themselves together, and then there are these chemotherapeutic agents, daunorubicin and doxorubicin, that also happen to fall into this cluster. And so my wife, who happens to be a cardiologist, noticed this figure. I was very excited. I said, oh, we made these nice figures. Look, they're so pretty. And she said, oh, that's really interesting from a clinical medical standpoint, because it's known that these chemotherapeutic agents that are used in clinical medicine have cardiotoxicity. And here we've, through a signaling assay and our connectivity map representation, linked them to a set of drugs that we know have cardiovascular phenotypes. We don't know what it means. Of course, it would require a lot of follow-up. But my conclusion from this is that even a cardiologist can understand targeted proteomics and the results that we get from this. So, okay. So the next question we had is, can the P100 assay be considered a Sentinel assay? And I just wanted to say a special thanks to Paola Piccati, who isn't here, but I see that Martin is here from her group. And I think this is just a great terminology, Sentinel assay. It conveys a lot about what we hope the assay can do. It's a readout for a certain type of biology. It's a readout for pathways that biologists will care about and be able to intuitively interpret. So we designed this following experiment where we took a set of inhibitors, kinase inhibitors, that are known to hit nodes in various signaling pathways: JAK-STAT inhibitors, MEK inhibitors, MAP kinase inhibitors, mTOR inhibitors. These are things that are all going to sound common, and importantly, folks who are interested in biology are going to be able to interpret the actions of these things. And then we measured with the P100 assay after either 3, 6, or 24 hours of treatment. In this case, I'm only going to show you data from 3 and 6 hours. And one of the first observations that we had is that the P100 assay can again distinguish between very close compound family members. Here, whether we took a pan cyclin-dependent kinase inhibitor like flavopiridol or a specific CDK1 and 2 inhibitor like dinaciclib, we could see that the signatures they developed could be easily separated using hierarchical clustering. They are similar, so that we could recognize CDK inhibitors as a class through the development of their signatures. But the strength of flavopiridol is quite different than the strength of dinaciclib. That could be a dose-related phenomenon. But at the same time, there are different probes in the P100 assay that distinguish them. We can see things that go down with flavopiridol that are neutral or go up with dinaciclib. And we can use the assay to distinguish these close compound family members. And the same thing with JAK2 and JAK3 inhibitors. The signatures that those things develop are quite different, and we can see, we can clearly separate those two things as a readout. The other thing that we could do is take those drugs that I showed you a couple of slides ago and project them onto the kinases that they inhibit. And then we kind of organize these just by going through the KEGG pathways and KEGG tables into a linear pathway projection. So here, each of the drugs is represented as one of these red hexagons down below, and I've simply drawn a line up to an oval, which represents the kinase or that it inhibits. And then, I had to fill in a couple of nodes along these pathways in order to make everything connect in this linear representation. But the general flow of this is from receptor, PDGFR inhibitor at the left side here, all the way down to things that affect cell cycling in the nucleus, like the cyclin-dependent kinases. And if we kind of take a step back, we can see that these drugs project onto kinases that roughly organize into 3 modules. The first is this MAP kinase signaling module, contains JAK-STAT, BRAF, and MEK. And then a PI3 kinase mTOR module, which of course contains mTOR, AKT, and so on. And then a cell cycling module, which contains the cyclin-dependent kinases and some things that are upstream and downstream of them, and we can kind of divide the pathway into these 3 logical modules. And we treated MCF7 cells with all these drugs. This is, in fact, a subset of the drugs that I showed you on a few slides back. And And then the first thing that we did is, without doing any clustering whatsoever, is just to look at the signatures that were developed along this linear pathway. So each major column now contains 6 different samples, because we did this in biological triplicate at 2 different time points for now. So each one generally follows the pattern of 3 samples that we measured after 3 hours of treatment, and then a subsequent 3 samples with the gold boxes above that we measured after 6 hours of treatment. And I think, without doing anything, but just by kind of observing this picture, you can see that the different drugs along this linear pathway projection tend to generate different signatures. Things here don't look like things here, don't look like things here. But the other thing that we kind of noticed by looking at these heat maps is that treatments that are proximal in the linear pathway projection tend to develop signatures that are somewhat similar to one another, right? The chance of these 2 neighbors here, these 2 neighboring drugs, having a similar signature, we thought, by looking, would be greater that these things are similar than, say, comparing something to over here. And then you can see here other compounds in this back end in the cell cycling module. They have similar but distinguishable signatures, at least by eye. So we decided to try to quantify this in a more formal way. And what we did is simply collapsed all of the replicates and measured, well, we measured the correlation among all the replicates and then collapsed those correlations by the drugs to which they were related. So we can represent that as now as a correlation plot, or a core plot, if you're familiar with that terminology. And so what we have is both now along the x-axis and the y-axis, just the list of drugs that are projecting onto this linear pathway. And here I've shown you the enzymes that they've inhibit down here along the side. And so everything on this diagonal is how well all the replicates of a given drug, whether it's at 3 or 6 hours, correlate with themselves. And what we can see is, for most drugs, there's quite strong correlation along this diagonal, which means that replicate recall, or the ability of repeating the experiment to generate a similar signature in the assay, is quite good. And the strongest correlation is along the diagonal. This is what we hope for and expect, and it's reassuring to see it. But what's quite interesting is the off-diagonal correlation. And so here you can see a kind of block of off-diagonal correlation that's really only among these particular treatments, among these drugs. A second block of off-diagonal correlation that relates to these drugs here, and a third block of off-diagonal correlation that mainly concentrates itself in this. And it turns out that these blocks of off-diagonal correlation almost perfectly correspond to the modules that we had just a priori determined that the different nodes in the pathway had fit into. And this was a big aha moment for us, because we hadn't arranged the pathway in this particular way to generate this effect, but we were very pleased to see that these blocks of off-diagonal correlation almost perfectly delineated the modules that we had divined from looking through KEGG pathway tables. And in fact, what we think is that the assay can reveal some modularity about the pathway. I see a question here. Yeah, I'll repeat the question for you.
How would you interpret the fact that 2 Jaffe inhibitors completely and now they don't correlate, and one clusters with the upstream and the other one?
So the question is, you have 2 JAK inhibitors, and a few slides ago I showed that they don't have the same signature in cells, and now one of them has a strong correlation, off-diagonal correlation with some of these things, and then one does not. And so recall that one is a JAK2 inhibitor and one is a JAK3 inhibitor. So in this case, it could be for these pathways, JAK2 activity is more correlated with passing the signal down the train than JAK3 might be for in this particular cell type. And that leads to something that we're going to talk about later about signaling in different cell types. So, good? Okay. Okay. All right, so now I want to show you a little bit about the project that we're working on, where we're developing a library of proteomic signatures and show you some real-life connectivity maps and some ideas about how to query these signatures. So I'm working on a project that's called LINKS. LINKS stands for the Library of Integrated Network-Based Cellular Signatures. This is together with the McCoss Lab at University of Washington and the Leeway Sci Lab at Massachusetts Institute of Technology. And this is what we are aiming to do to create this proteomic signature compendium. We're taking various cell lines, some of which you've already seen, but now more, and also neural lineages that we're differentiating either from ES cells or from iPS cells derived from patients. And we're treating them either with drugs, manipulating their genes, or driving neural lineages down certain fates, which is another type of perturbation, bringing a cell from one state, one fate, or one stop along the lineage train to another. And now we're measuring with both of our targeted proteomic assay panels, our P100 phosphosignaling, and we're doing this in a way that's kind of a hybrid DIA PRM assay, which I don't think I'm going to have time to talk about, as I mentioned. We're measuring chromatin profiles from these same treatments, and then we're also measuring a sort of proxy of expression profiling. So we'll have all these types of data from the same treatments and the same types of cells, and what we hope to assemble is this compendium of first signatures, use them to formulate connectivity maps, as I've shown you, and then try to figure out the circuitry, perhaps suggest some cause and effect of how signaling and epigenetics interplay with one another, how changes in signaling might affect changes in epigenetics and vice versa. And so what we're doing is we have 3 different sets of small molecules. Some are epigenetically active, some are active in signaling pathways, as you've seen, and then some are very specific, looking at pathways that are active in neurodevelopment and neuropsychiatric disease. And then we want to correlate these to disruptions of genes that are either associated with epigenetic processes or neurodevelopmental processes. And so over the course of 6 years, and we're about a year and a half into this, we hope to generate over 10,000 different biological perturbations, resulting in really what is 30,000 samples when you look at the 3 assays. So 30,000 profiles, collapse those into signatures, and then use that as a sort of big data set to try to infer something about connectivity and circuitry. So now I'm going to show you some data from our early efforts here. All these data will be looking at a series of drugs that are known to have epigenetic activity, as well as some control drugs. But the first question that we needed to ask is, with these new drugs that the assay was not necessarily trained on, neither assay was trained using these drugs for the most part, would signatures be able to be generated that were reproducible across them? So luckily, the answer is yes. First, we can look on the left here, look at correlations of signatures in the chromatin assay. So of course, these drugs are epigenetically active. If we didn't get this one right, we were kind of dead from the beginning. But what's kind of reassuring here is if we compare the correlation of any one sample with any other sample, and we do that for all samples, and plot the distribution of those correlations, what we see here in this pink distribution, which is just sort of like a smoothed out histogram, is very nice kind of null distribution centered around a correlation of 0 and with a spread from about -0.5 to 0.5. So no drug on average is correlated more or less with any other drug, at least through the lens of epigenetics. Then if we look at replicates of the same compound in the same cell type, now that's this green distribution, we can show that the correlation distribution is significantly shifted towards a much higher correlation. So this is good. Replicates recall themselves or are more similar to themselves within a cell line. And then we can look now at this intermediate distribution, this purpley distribution here, which is the same drug, replicates of the same drug, now compared across different cell lines, because the biology might be different between cell lines. These things might not work the same in different cell lines. And at least in the epigenetic assay, what we saw is that largely the compounds work the same way among different cell lines. If we look at the phosphosignaling, however, we see something that has one key difference. Again, the null distribution is centered around 0. No particular treatment has any given affinity for any other treatment on average. Replicates within a cell line are now shifted towards higher correlation coefficients. Now, the thing that's important here is that these are drugs that we knew would have epigenetic effects. We weren't sure if they would have any phosphosignaling effects at all. If they didn't, everything would just fall into this central distribution of no correlation. But that we were able to observe a shift in the correlations towards a higher correlation coefficients sort of gave us a clue that these epigenetic perturbations do in fact have some consequences in the realm of signaling. And then if we looked at replicates of drugs across different cell lines, we saw only a very modest shift in the correlation coefficient. So this gave us a hint that maybe something was unusual here, and we'll talk about that more in a moment. Okay. So of course you can't really see anything on this map, but it's just meant to show you that even from these now very large scale data sets. So this is 5 different cell types, 32 different compounds, each done in biological triplicates. So about 500 different samples. We can formulate these connectivity maps here at the beginning. And so what I'm going to do is zoom in on different segments of it, starting with this upper right. So we saw this self-organizing cluster. Again, now I have colored the centers of the nodes with and I've even labeled their drugs and used different shades of green here for all of the HDAC inhibitors. And you can see that all of these HDAC inhibitors self-organize in our connectivity map. The outer rings, again, represent the cell types, and so regardless of cell type, they're generating the same sort of signature in the chromatin space. And then, unfortunately, it looks like the lines are a little bit washed out here. Yeah, it's hard to see there. But in fact, there are some other compounds that are not HDAC inhibitors that also connect to this self-organizing cluster on the map. Methylstat affects methylation of histone proteins, not acetylation of histone proteins, but we might infer from this connectivity that there could be some crosstalk of this drug or some off-target effects of this drug that does play into the acetylation pathway for histones. And then we can look at a series of compounds that now target EZH2. Again, these things all self-organize into one segment of the map and maybe start to observe some additional connectivity of drugs that don't necessarily target EZH2, where we can follow up and see if these things are in fact generating chromatin signatures that would be more consistent with EZH2 inhibition than whatever its intended mechanism of action is there. And then we can observe new clusters of biology, things where we didn't quite know what was going on from some of the drugs that are not necessarily chromatin-active compounds. Actually, I misspoke. This is 64 different compounds. This includes some of our neuroactive compounds here, so it's even more. It's probably around 1,000 samples or so here. And what we can see is this kind of new cluster that contains the drug resveratrol. Resveratrol has been a very controversial drug. It's thought to activate a protein called SIRT1, which is important in histone acetylation. But interestingly, it clusters with another drug called trichostatin A. Now, trichostatin A is very commonly known as a histone deacetylase inhibitor, but at least in a few different cellular contexts, it appears that it's not clustering together with the bulk of histone deacetylase inhibitors. And if you look carefully here at this first map, there are a couple of instances of trichostatin A behaving like an HDAC inhibitor in say, PC3 cells or in A375 cells. But in other cell types here, trichostatin does not seem to behave as an HDAC inhibitor. So you start to reveal some of the cell-specific biology of the mechanism of action of these compounds that we can get through our proteomic signatures. And then, of course, it's very early to start interpreting the phosphosignaling data, but we can kind of see very similar things starting to emerge in phosphosignaling, even though we're looking primarily here at epigenetic drugs through these initial data. And here we can see, interestingly, that trichostatin A does, at least from a phosphosignaling lens, self-organize together with other HDAC inhibitors. So the fact that the HDAC inhibitors have any phosphosignaling consequences at these short time points at all is sort of surprising. But at least through the lens of phosphosignaling, trichostatin is doing what it's advertised for. And so we can see some early structure emerging through the phosphosignaling data here, and we look forward to building out this collection a little bit more. And so just one more thing about these data is again, I've sort of been alluding to this fact that there's cell-specific biology, and then there's biology that transcends cell types. So we try to quantify this in some sort of way. So if we take those maps that I just showed you and organize them in a slightly different way, what I've done is grouped all of the nodes by their cell types. Here I've limited it to 4 of the cell types here. And then I can draw a line between any 2 given nodes when there is a strong correlation between them. And I've color-coded those edges by the type of assay through which the connection was derived. So anything that's a blue edge that goes between different that goes between different nodes is derived from the chromatin signaling assay. Anything that's a pink or a red edge is derived from the phosphosignaling assay. And what we noticed, there were a lot of blue edges that were moving between cell types. And there were a lot of pink or red edges that were sort of staying within a cell type. So what we did is we added up all of the weights of these edges for both the chromatin connectivity, or the blue edges, and the pink ones from the phosphosignaling. We saw that about 60% of the edge weight in chromatin moved between cell types, while about 60% of the edge weight in the phosphosignaling stayed within a cell type, such as to kind of get the idea that no matter what you do, epigenetically, the result is going to be very similar regardless of cell type. But cells respond through signaling in different ways that is very specific to their own biology. Okay, so let's see how I'm doing on time here. Okay, so I want to talk about one more thing, and this is about query. This is about moving beyond heat maps and even moving beyond connectivity maps, which are just visual representations of how we can try to intuitively interpret these data. But what we really need is a principled way of telling what looks alike with respect to an assay signature or a profile in a targeted proteomics assay. Because as I was sort of saying, staring at heat maps really only gets you so far in these things. And we want this method to be generalizable for looking at multiple different assay panels and even extend perhaps beyond proteomic data into transcriptional data. And we also want a way to prioritize our follow-up strategies. What are the first hypotheses on which we should follow up? And so you could think about how you might use such a tool, such a query tool, from a variety of different standpoints in biology. If you're a geneticist and you could look up through profiles connections between drugs and genes, you could find pathways that were activated or deactivated by certain genetic lesions, again, using our compendium or library of signatures that we hope to build. If you're a cell biologist or a biochemist, you could hope to find within our assays proxy biomarkers of either drug efficacy or proxy biomarkers of disease or processes disrupted in disease. You could discover crosstalk between drugs and how they might impinge on certain cellular signaling pathways. If you're a chemist, you might be able to use a query mode to get some insight into the mechanism of action of a compound by comparing profiles of compounds that you don't know what they do to compounds that you do know what they do, the kind of guilt by association. And if you're, say, a drug hunter or looking for new leads, or you're a physician scientist, you could try to find drugs that generate profiles that are perhaps anti-correlated with profiles of disease states or known perturbations that would, say, induce a disease or models of disease. And then, of course, you can characterize new genetic lesions or new environmental insults into cells and try to find drugs that will either help you model them or help you counteract those perturbations. Okay. And so we use a KS statistic framework in order to execute these sorts of proteomic signature queries. And so we ask kind of three different questions. We start, as before, by calculating correlations between the z-scores of all of the replicates for all of the analytes. And then we simply want to look at the distribution of correlation scores or connectivity scores from samples derived from one drug versus all the other samples in the entire data set. So then the second question that we're going to ask is, where do the replicates of that particular drug perturbation fall within the overall distribution of all possible correlations? And this gets towards replicate recall, or whether there is a signal for that perturbation in the assay. And the third thing that we can ask is, what other drugs have outlier correlations with a drug that we're particularly querying? So I'm going to illustrate this through the drug pazotinib. And so if we just plot now in the P100 assay, all of the replicates, or the correlation of all the replicates with all of each other, we see a nice distribution here. On average, the correlation is about 0, and then there are some things that are way out here. And so now we want to ask, where do the treatments, the drug replicates of pazotinib, fall with themselves? And so we take this histogram and we kind of smooth it out into what's called a violin plot. And then I add little tick marks onto the distribution where we have replicates of pazotinib. And here we have three different types of replicates, ones that are compared within the same time point at 3 hours with the red tick marks, at 6 hours with the blue tick marks, and in green for comparisons that span across different time points. So that's we can ask the question, is the same signature essentially present, or its signatures correlate across different time points of treatment? And so these things started out as being called violin plots, but I've put little frets on them, so now I call them banjo plots, by just trying to show where the replicates of the drug fall within this distribution. And I think you can appreciate that all of the replicates fall towards the right side of this distribution, or skew towards higher correlation coefficient. And so you can develop a statistic, or you can ask, what is the KS statistic for the distribution of these replicates versus the distribution of all of the potential replicates for this given drug? And you get a statistic out of that, and we can use that to help rank order our query. So you can compute this KS score, you can compute a p-value from this. In our case, I think we need to work on the statistics of computing p-values of the KS statistics, because the p-values that we're getting right now are all incredibly significant. We understand why, and so we're working on resampling and permutation methods that are going to allow us to get some more realistic probabilities associated with things being connected here. And so we can now look across that entire set of kinase inhibitors that I showed earlier and generally ask the question, do samples correlate well with themselves? Do replicates correlate well with themselves? And I think you can probably see that for most of these distributions on most of our banjos here, all the frets are out at the right end of these plots, which indicates good replicate recall, and that there is signal in the assay. Not every drug generates a strong signature. Not every drug recalls itself well, and that's just life, but that's why we do the experiments, right? Okay, so now we can ask, if we query a drug, not just versus itself, but versus all the other drugs in the set, how do the replicates fall with respect to correlation? So here I'm showing you a query of dinucyclib. And what I've done here is ranked it by the KS statistic in a sort of a special way, where we take the magnitude of the KS statistic and then leave it positive if if things are to the right of the median of the distribution, and make it negative if the tick marks are to the left of the distribution on average, or actually on median. And here, if we query Dinocyclib, we can see that the top ranked hit is in fact itself. Good replicate recall, sort of good condition for entry. And then the second thing that we see here is flavopiridol, and you may recall that flavopiridol is also a CDK inhibitor, right? So now we're able to find compound class members through this KS query framework. And then we find staurosporin, which is interesting, but as we know, staurosporin does a lot of things. And then the fourth drug down the query list is this SCH 90776, whatever that is. But it turns out that SCH 90776 is a CHEK1 inhibitor. And if we go back to our pathway projections, we can see that CHEK1 does feed down into signaling and control of CDKs. And so quite nicely, the query has recapitulated this little bit of this signaling network, and we can infer that these things might be working in some sort of coordinated way. And then we can do this also not only for phosphosignaling, but we can do it for chromatin profiling, and we can use these statistics to play some interesting games and go hunting for hypotheses. Again, now I'm showing you many different drugs, and just looking at their own replicate recall. Again, things are generally kind of finding themselves, not in all cases, but in many cases now using chromatin signatures. And then we can start to ask, what about some of those drugs? So we can look at GSK 126, which is an EZH2 inhibitor. It finds itself most strongly with replicate recall. And then the next two things that it finds are two compounds that I've highlighted here with asterisks. They are also EZH2 inhibitors, so we're able to stratify these things into the same class. And then there's the next most significant hit is this curcumin drug, which is not known to be an EZH2 inhibitor. So the first thing that we did is, okay, let's make the reciprocal query. Let's ask, how do curcumin signatures line up with other drugs? Because just because there's connectivity in one direction doesn't mean that there might be in the other. But in fact, the top three hits beyond itself are all the EZH2 inhibitors for curcumin. So we're starting to go down a path and say, well, maybe there's some subtle off-target effect of this compound, which does many, many things. Curcumin is actually the yellow compound in turmeric, and it's got a variety of potential human health benefits that no one quite understands how they all work, but it's been used as a homeopathic remedy for many things, and has been even investigated clinically to a certain extent. And so maybe we can use our assay to start to infer that maybe it's got some kind of effect on epigenetic processes. Now the nice thing is we have put all of these data into a Panorama repository that we have set up for our links project. So you could go and download all these data, these same data that I've been working with, the entire skylines. You can see our entire targeted assay panels for both the GCP and the P100 assays. And then what we've built is one special module in Panorama that lets us recall subsets of these data and plot them in heat maps. And so if you went to our Panorama repository and said, create this custom GCT, which is just a heat map with some annotation on it, I wanted to see how strong was the effect of the signature of curcumin compared to these other drugs. And so I simply asked for the results from a set of different drugs, and could get back in very rapid fashion this heat map here. And what I've done now is highlighted all of the curcumin replicates here. All of the replicates over here are all these EZH2 inhibitors. You can see they have quite a strong signature. In fact, curcumin doesn't have such a strong signature that mirrors that of EZH2 inhibitors. And so we're not really sure if we're onto the right thing here. But at the same time, our ability to kind of seamlessly go back and forth between a universal query framework and being able to look at and interact with the primary data really helps us to use targeted proteomics assay panels in ways that I think geneticists and biologists would come to expect. And up to this point has been quite difficult for the proteomics community to kind of leverage into simple and intuitive tools. Okay, so I'm just about done. As I mentioned, this links project is a public effort. You can go to our Panorama repository, the address is here, or you can just Google links Panorama, and it'll get you there. When you're there, you can learn more about the project. There's some more about the background of the project, the drugs that we'll be profiling, and the genes that we'll be profiling. You can download all of these data, including the custom subsets that I show you, and then you can interact with data. There are links from our data sets that you can instantly get a heat map that you can interact with and cluster and do your own analysis or whatever you want. And so I just want to leave you now with 3 key points that I think are kind of important, and I hope that many of you will agree with them. I think you probably wouldn't be here today if you didn't agree with them already. Proteomics can illuminate areas beyond the reach of genomic techniques, things that you just simply cannot read out with nucleic acid techniques. And this is extremely important. It's not to say that transcriptional profiling and DNA-based techniques are bad. It's just that proteomics can add complementary information to them that is just simply inaccessible through those techniques. Importantly, targeted proteomics is the paradigm that I think will let us play nicely in this world of high throughput genomics and transcriptomic analysis, because we can make our unholy data. And I strive to be as unholy as possible in all of my scientific efforts. And then we have now this concept of proteomic fit-for-purpose assay panels that are good for generating signatures. They're useful for biologists. They can intuitively interpret what's going on by comparing like with like and noting contrasts between different things. And we can now use these frameworks to develop sophisticated analysis for today's kind of big data environment, including all the things that I've shown you examples of, and clustering, connectivity maps, and query frameworks, but also things that I hope folks will interact with these data and develop on their own. And so with that, of course, there are many people to thank at the Broad Institute, especially Jennifer Abelin and Jinal Patel for helping to develop the P100 assay panel. Amanda Creech has been very instrumental in solidifying our chromatin profiling, and you can see her paper in methods from last year. The entire team at University of Washington, which includes Brendan and Vagisha Sharma for the Panorama repository, collaborators really all over Boston and all over the country, both in academia and in industry, that have allowed us to move these studies forwards. And thank you very much for your attention. Happy to take any questions.
Question about the methodology.
Mm-hmm.
How did you normalize across your batches of 96 samples, and how did you normalize across in time the samples you did, separately? So.
Yeah, it's a good question and has several different answers depending on the assay. So the histone assay, it's pretty simple. First, we normalize for load by taking a histone peptide that is not commonly modified. So that means regardless of the amount of histone that we load in there, it should be a sort of constant thing. It shouldn't get modified in different ways. It shouldn't change in funny ways. So we use this as a kind of loading control, like a loading control in a Western blot, and first normalize all the histones in each sample to that particular peptide. And then what we do for each probe is we median row normalize within a batch. And we develop z-scores within that batch. Since each batch usually is within a single cell type, we think that this makes the most sense. For the P100 assay, the load normalization is a little bit more tricky. And what we do is try to fit an average model of profiles across all the samples and bring those to some theoretical median looking profile and minimize the distance between any one given profile and this theoretical median profile. And so we call this load balancing. I can tell you more about that offline. It's a little bit subtle. And then we do also median row normalization and convert everything to z-scores to kind of account for the variability of different probes.
Two questions. First one is about the histone profiling. So the data seems of really high quality and consistent, and this makes it kind of ideal for PTM crosstalk. Have you looked into something like this? And then the second thing is related to data quality. So when you have such large-scale studies, how do you implement or plan the concepts of blocking and randomization or things like this?
Yeah, so every single replicate we have is a biological. I'll answer the second question first. Every single replicate we have is a biological replicate. We try to analyze things in a way such that direct biological repeats are not organized next to each other, or not analyzed right next to each other. But we also need to be kind of systematic and help our technicians and analysts be able to execute the upstream preparation of these samples in a routine way. So the way that we organize the plate is to have replicates next to each other in rows on a plate, and the way that we analyze the plate is to go down columns so that there's a periodicity of, say, 8 before you encounter a different biological replicate. But I know, and from having heard Olga's lectures in the past, and you're about to hear Olga's lectures next, that there are smarter ways to do this. But we also need to have a balance between being able to execute our workflows in an error-free way and being able to at least do something that's not going to bias our acquisition too much. So. So the first question you said about PTM crosstalk, do you mean specifically with respect to histones?
No, specific modifications going up and down because you have a lot of systems, you perturb them, and you have the same readout, which is a methylation, dimethylation, different PTMs. So you have a lot of data points on the same PTMs, on the same sites.
Yeah.
So you will be ideally, you should be able to see how they crosstalk to each other, how they behave across the cell lines, and not how the cell lines behave across the PTMs.
Yeah, no, absolutely. And actually, it sounds like a great question, a great direction to go. And I would say if you're interested in that, please go and download the data and let me know what you find. Maybe you'll find something amazing. But just let me know.
You mentioned that there are hundreds of thousands phosphorylation sites measured, and when you went and measured yourself again, so could you please explain what was the rationale for this, to select this P100?
So there have been hundreds of thousands of phosphosites reported. What we needed to make is a systematic perturbation of systems to try to determine which phosphosites would be most responsive to drug or other sorts of perturbations. Just because you could look up in phosphosite.org the 100,000 phosphosites doesn't mean that there's quantitative data about all of them, or that you could easily do a meta-analysis of different perturbational studies of all of those 1,000 or 100,000 phosphosites. So we did need to make some of our own data so we would have a common starting place with which to distill down from the 1,000 or so that we started with down into our 100 that we measure. We also wanted to enforce a couple of different conditions, namely that we didn't want to have to do a lot of fractionation upstream to get to the assay. We wanted the assay panel to be compact. The whole assay takes about 80 minutes from inject to inject, the entire gradient is only 60 minutes. And so we intentionally biased for lower hanging fruit, meaning things that are of slightly more abundance that can be seen in a single shot proteomic experiment and that were commonly observed across multiple cell types. Just because a phosphosite has been reported somewhere doesn't mean that all the metadata and annotation and the conditions under which it was observed are well reported with it.
Hi, great talk, thanks. So this P100 assay, you also call it DIA. So what's the DIA in there? Do you kind of have an inclusion list for those 100 peptides or phosphosites you want to look at? And do you cover the remaining M over Z range with the rest of the time that's left over, or what's the DIA in there?
Yeah, so good catch. When we initially proposed this experiment, or this project, is we proposed we will measure all of these phosphosites using our PRM assay, And then we will take some of those samples or all of those samples and also acquire DIA data on them all, because we've done this phosphopeptide enrichment. It would be desirable to see all kinds of stuff in there. And so what happened is the funding agency came back and said, well, we're going to approve your project, but we're not going to give you all the money that you asked for. So what we did is we have kind of morphed the assay into this hybrid PRM DIA assay, where the data are all acquired just using a DIA or swath, or I just like to call it comprehensive proteomics methodology, where we're rastering windows across a mass range. But we still have synthetic peptide internal standards for these 96 analytes that we care very deeply about. So we do true ratiometric quantification on those analytes, even though the data type that we're starting from is comprehensive proteomics. I can tell you that the PRM assay version of it is cleaner, the data are less noisy, it's more sensitive, all the things that you would expect when you're contrasting a PRM assay that's dedicated for certain analytes to DIA assay. But the DIA assay does a pretty good job. As you've seen, we can develop signatures and formulate connectivity from those data. I fully am aware that the data are not as high of quality as a pure dedicated assay, but now I'm excited to start to look at the DIA data and query them for other analytes. There should be thousands of other phosphoanalytes that we can see, and in a way we could have virtual panels. Right? You could make up whatever set of P100 you wanted to look at, ask the DIA for those peptides, and say, well, are there interesting signatures here? The trick is going to be to get good quantification on those. And what we want to be able to do is leverage these 100 phosphopeptide internal standards to inform the quantification and normalization of samples to one another across this, what will ultimately be a very large data set. But yeah, great catch.
So another question on the P100 panel, I guess. So the way I understand what you did in the first step to get to this list, you measured 1,500 phosphosites. You weeded out everything that doesn't change, but you probably also collapsed things that are redundant. For example, if you have a certain set of phosphosites look the same across many conditions, you pick one of them. Is this
That's right.
Yes. Yep. I'm wondering whether now you can use this and to reverse what you have done to, for example, predict the phosphorylation state of a certain protein based on, let's say, drug treatment based on these set. If it works, it would be very great.
Yeah, no, that's another great point. And in fact, that was our original aim, was to be able to infer the set of 1,000, and we generated an imputation model, inference model for it to do that. But then when we kind of had this condition of, well, combine the DIA and the PRM assay into one, we thought that sort of goes away, because now we should just be able to look up those 1,000 in the data themselves from the DIA data. But yeah, it's a great idea. It's what we're starting from.
So maybe a quick follow-up. Can you actually use this to now, from data, for example, let's say you knock out different kinases one at a time, or knock down different kinases one at a time, actually to infer back the phosphorylation network? Because at the end of the day, what we're interested in as biologists is typically something. It's like what are the targets of a certain kinase, and if we can look at 100 and can infer from this. For another 1,500, the targets would be very useful.
Yeah, I guess in answer to your question is I have the same exact question, is can we do this? Can we systematically generate enough perturbations so that we can understand the networks and the control circuitry? And I think we don't have enough data yet to even be able to answer the question of whether it will work. But I would, if you have ideas, I would love to, again, talk to you about it. And yeah, it's fantastic.
Hey Jake, I have two questions. So you made these two different assays, one to profile chromatin structure and one for phospho, the P100, and you also applied both on the same samples, right? These were these two on this very big data set that you showed. So what I would expect that this chromatin change also influences phospho. And did you, for example, do an analysis where you checked whether you get similar clustering Because you always show that you can cluster certain conditions together, and do you get similar clustering from phospho as well as from chromatin analysis or not? And
Yeah,
what's your
so
thinking on that?
the answer to that is sometimes yes and sometimes no. And I think that the sometimes no are the most interesting cases, or something like where you could see that maybe two classes of compounds are correlated positively in the chromatin space, but anti-correlated in the phosphosignaling space. And so there's a postdoc in the group, Ryan Peckner, who's where's Ryan here? He's a mathematician, and so he's asking these very questions about what are the general properties of correlations within assay and between assays, and also with transcriptional assay, because sometimes we'll see something like a chromatin assay and transcriptional assay agree on consequences of perturbations, whereas the phosphosignaling might disagree with those two or something else. So those are the interesting in-between cases. Did you have a second question as well?
Yeah, and one technical one. So you do now PRM or DIA assays, whatever. The analysis software is Skyline, right? You do all the analysis in Skyline. Do you use the automated function in Skyline, or do you still go manually through all your data? What's your data analysis strategy?
Yeah, right. We start with automated and then manually review and correct. It's in DIA. It's challenging to not have to review everything. At this point, we're reviewing. For the chromatin, the assay panel is compact enough that Skyline is almost always picking things correctly on the first try.
But you think it's also because you have the heavy reference, or a reference?
Well, we have
pattern
heavy
in-
reference in both cases, but yeah, DIA data is just still challenging. I'm quite confident that instrumentation advances will make comprehensive proteomics really the default standard that people will do in the future. I think the trajectory of those advances is clear. It will just take some time. But nonetheless, we're making a bet that software geniuses will be able to extract all of the signals that we really want to get out of these data. So we're collecting the data prospectively in this way with the hope that we'll be able to deeply mine them later. So, yeah.
Maybe it's just obvious for everyone, but not me. But how do you correct for the fact that the abundance of your protein in this P100 might be changing and the results in a different correlation level is just a result of that?
So what I didn't tell you is that we're measuring the phosphosignaling changes at a pretty early time point. We measure them at 3 hours. We measure the chromatin changes actually at 24 hours. Lots of chromatin changes are dependent on a cell cycle or something like that. But so we felt like at 3 hours, the changes in overall protein levels would be relatively small compared to the changes in phosphosignaling, which tend to be relatively quick in terms of the time scale at which they occur. But the second part of the answer there is that we accept that fact that there may be protein changes. Nonetheless, what we're reading out is a change in the total level of that phosphosite, one way or the other. And the fact that that level is changing in a reproducible way probably indicates that it has some meaning or some consequence in the cell. Yeah.
Okay, thank you very much. We'll have a short break now for 15 minutes, and then we continue with Olga Witek's talk.